Welcome back, discoverers. Today, we have two incredible guests exploring different angles of gene editing and delivery.
We are starting to see a shift away from early generation particle characterization techniques. We're seeing a lot of different technology.
There is evidence that it might be useful also to kill cancer cells.
Yes, today we will dive deep into both sides of the gene editing coin, from the molecular scissors that we all have heard about in CRISPR to delivery dashboards. That sounds kind of like DoorDash for gene editing, right?
Well, we'll find out on this episode of Discovery Matters. Connor meets Stephan.
So I'm Stephan Riesenberg. I work at the Max Planck Institute for Evolutionary Anthropology.
Max Planck Institute, yeah, I've heard about that place, but evolutionary anthropology? That doesn't strike me as very Max Plancky.
Well, that part of the institute was founded back in 1997 with an aim to bring together scientists from, and buckle up here because this is some exciting cross-disciplinary collaboration here.
And we love that stuff on this podcast, don't we?
Genetics, primatology, psychology, linguistics, and other fields, all to study human evolution.
That is just fantastic. So where does Stephan fit in here, and what part of our evolution is he looking into?
I'm a group leader in the genome engineering and repair group, and we mostly work on making editing more efficient.
Part of making editing more efficient is done using something called synthetic guide RNAs.
So if you do CRISPR editing, you need, of course, the CRISPR protein, but also an RNA to guide it there. And in the past, people mostly used plasmid-derived guide RNAs that are produced from plasmid. But we use synthetic guides because they're generally more efficient and ready to use if you buy them.
Stephan and his team are essentially then chemically recreating RNAs instead of needing the biologic RNA that's created by a living thing. Is that right?
Correct. And one of the main things Stephan uses in his work is something called the EVA score.
I'm guessing that EVA stands for Evolutionary Anthropology. Is that right? Like the famous score from MIT, which is called the MIT score?
Yeah, that's the one.
Okay, so not off-target predictions, but in this case, what does it tell us?
So the score informs you if a guide RNA, a synthetic guide RNA, is likely to cut or not. And to simply say if the score is below 50, it's a bad guide. If it's above 50, it's a good guide. But surprisingly, most of the guides are actually good. That are synthetic guides. So if you have 93% of guide RNAs actually have activity of at least 40% when they're synthetic in our data set.
And the problem Stephan first set out to solve that led him here is what exactly? And how then does synthetic RNA and CRISPR working together start to help him solve that problem?
Stephan looked at other CRISPR users. He noticed how they had problems with efficiency. First, they needed to pre-test guides, and then the prediction tools that were available to them were trained on transcribed guides. So he just wasn't getting the depth of information that he needed.
And so there was a discrepancy. And so we wanted to solve that. And we had two main ideas why there's a difference. And first, that if you have a transcribed guide, you have a promoter bias, the polymerase which needs to make the guide. So the sequence of the guide maybe has some influence. But if you produce it synthetically, it's very normalized. It's just you add one base after another. So there shouldn't be this bias. And also the metric to determine guide RNA efficiency previously was mostly indels, so short insertions or deletions that are formed after a CRISPR cut. But this is actually not the only thing that happens in the cell. The cell can also die as a consequence. It can repair it perfectly. It can have other effects. So we also thought just the metric of indels is not good enough. And then you won't often underestimate.
So, listeners, I had to look this up. Indels is not incels. Indels are basically another way of just saying insertion and or deletion of nucleotides into the genomic DNA. It's a shorthand for gene editing by inserting or removing.
So you can think of a scenario that you edit your target, and let's say 80% of the DNA is actually cut in the cell population. But 60% of them are dying. And the rest, only a few actually make these indels. And the readout in the end is you extract DNA from the surviving cells. You see you have little indels, so you think your guide actually didn't work that well. But you didn't see what happened before, that maybe most of them actually died.
And the work that Stephan is doing has different dimensions.
So first, it solves a long-standing problem that you can actually predict if your guide works, if you use synthetic guides. So you don't need to pre-test several one of them. So you save time and costs. But also what is maybe more relevant in a therapeutic or clinical setting is that we found that if you use a Cas12a, it makes much less large-scale unintended side effects than Cas9, at least in our data set. And of course, unintended outcomes are very bad for therapy. So the data suggests that maybe Cas12a may be a safer method.
And our super famous revolutionary enzyme Cas9 that we've talked about before when we talked about CRISPR, sometimes referred to as the genetic scissors, is basically the everyday workhorse. Mm-hmm. And we've heard a lot about it because Jennifer Doudna and Emmanuelle Charpentier won the 2020 Nobel Prize for chemistry in developing it. But Stephan's looking at a different molecule here, a different enzyme. Is that right?
He started focusing on Cas12a, which is more amenable to the work he's doing with editing genes. Stephan himself thinks of the 12a as the second place CRISPR tool after Cas9.
So if stuff doesn't work with Cas9, people often use Cas12a. Also because it has a different requirement of a PAM, which is a short sequence next to the target site. And so it gives you more variety where you can actually cut. And it makes a staggered double-strand break, and Cas9 mostly makes a blunt one. So there's some technical differences.
And Stephan must have an insight into the clinical applications or otherwise and the impact on CRISPR research that this is all going to have.
It is, of course, hard to say. But that there's a tendency that Cas12a makes less of these large-scale unintended effects that are harder to detect makes it, of course, a more promising starting point. I wouldn't say that, of course, that never happens. I wouldn't say that never happens with Cas12a, but it is good to start with a system that has a lower tendency for these effects that are hard to detect.
Long-term goals are most important. So what does he think those are when he thinks about his research over the next 5, 10, 15 years?
Pretty clear and simple. He wants to continue to make genome editing more efficient and easier to use.
One thing is we are working on a startup, ClearCut Biolabs, and that is aiming to provide genome editing kits that you have like efficient editing, reliable results, faster turnaround time. And in the academic lab, I can tell you some examples. So we have one exciting project where we developed a selection system that can select cells based on DNA sequence and not with antibiotics, which could be very useful in cell culture, but also other things. When you have edited a cell and only a few cells in your population of cells are edited, what people typically use is put in an antibiotic resistance gene in addition, or sort with a fluorescent protein. But this is not, you cannot do that with point mutations. And also it's not natural to have this in the cell. And what we have developed is a method that just based on one nucleotide difference, you can select a cell. So you can kill cells that you don't want.
Ultimately, this ensures that what clinicians might put back into a patient contains only the modified cells, and that directly benefits the patient.
So the most immediate use is in cell culture. If you engineer or want to model diseases in cell culture, But there is some evidence that it might be useful also to kill cancer cells. Not really edited cells, but cancer cells. But we just did cell culture experiments. Of course, it's kind of a far away. But it could is a promising avenue, because cancer cells, by definition, have a different genotype.
It's so fascinating that if we're really to make a breakthrough on, our global cancer moonshot, because everybody's having a crack at it, so many different branches of science have to pull together. And what Stephan's doing here is potentially going to provide really important new insights into how we might ultimately, wrest cancer under control, defeat it, make it, permanently a manageable disease.
Yeah.
Does he believe then that this is achievable in our lifetimes?
I think for some types of cancer, not necessarily with what we do, but there are many promising avenues. I think also with these T-cell therapy, not all of them, but some maybe.
Now, his work doesn't just stop there. Stephan is in the midst of some other really exciting projects.
We have kind of solved editing for a single gene, at least in cell culture. That can be not done very efficient. The next goal is to do efficiently many different edits. And so we work on multiplexed editing of many genes at the same time. And one example we do here in evolutionary anthropology is revert human stem cells back to the ancestral state of the common ancestor of human and Neanderthals. And then in a cell culture dish, we can investigate some molecular phenotypes. And we have done that for several genes, but it is, of course, a big goal to edit many of them and then check how they work in interaction.
And the purpose of this, he says, is to learn all there is to learn about what makes the modern human on a genetic level.
Because our ancestors are extinct, there is no comparison really. But with CRISPR, it's a tool, one can ancestalize them in cell culture. So it's not to make an ancestral human, it's just cell culture to look at maybe how metabolism changes. What makes us human? This question is almost a philosophical one. And there are many, many factors. And genetics is a part of it. And everything we can find is only some endophenotype in the model. Of course, you cannot compare the diet. But it still brings us a bit further in understanding.
To be clear, Stephan does not think that base editing and prime editing per se are dangerous. He stressed that genome editing as a whole can be both dangerous and useful. Base editing and prime editing are not necessarily more dangerous than other genome editing tools.
And what about the future of gene editing? I mean, we've gone from Cas9 and now Cas12 and all sorts of new scissors. So where are we going next?
I think what's already very exciting is this CAR T-cell therapy, and they're also editing can help to make that more efficient. But I think I'm also very excited about this base editing or prime editing. But it's also a very dangerous thing because one can potentially do many things with it. But I think it's important to investigate it.
Okay, Dodi, I'm sure we've got another guest. Who is it?
Well, if Stephan is improving the genetic editing tools themselves, our next guest, Adam Crowe, Senior Manager at the Nanomedicine Analytical Development Team at Cytiva, is optimizing the vehicles and dashboards that take those tools into the body and tell us what they're doing once they get there. So we turned to a colleague for help.
So I came to what was formerly Precision Nanosystems, as now the Cytiva Nanomedicine Group, back in 2018, straight out of a postdoc in Vancouver. I was obviously looking for new opportunities, and nanoparticles offered a really unique skill set in which there was an analytical background, a chemistry background, formulations background. So it brought many different facets together. And what was really attractive to me is at the time, I think I was the fourth person hired onto our services team. And it gave us the ability, or gave the team the ability to really put a stamp on what the future of the services side looked like.
Right from the start, Adam was shaping the way his team would approach LNP analytics.
I was very fortunate to have the opportunity to build up an analytical team, bring in equipment, bring in associates. And I think we've been very successful on that and becoming an industry leader in analytical development for LNPs.
And how big is Adam's team now?
So the preclinical team of which we sits at around 20 people, something like that, and that splits between formulations, analytical development, and bioassay development.
Much like Stephan's team in Leipzig, Germany, Adam's team is dynamic, bringing together all kinds of skills.
I think the best way to put that is we are dynamic in that we respond to the needs of our client, the needs of the industry. Initially, so myself, I come from an enzymology, mass spectrometry background, and I came with that skill set. The first hire I hired was a material chemistry background, a physical chemist. And since then, we've really gone back and forth. We've had more mass spec people, mass spec specialists. We've had additional chemists, enzymologists, engineers, always looking for someone who has a different way of thinking than myself, because there's no point in duplicating yourself.
And are LNP analytics particularly tricky when you compare them with, for example, older ways of formulating biologics or even small molecules?
Adam describes LNPs as offering three major challenges.
So the first of which is they are a multi-component system. That means you have your payload with all the complex analytics with it, typically mRNA, but you put a lot of things there. And that could list off probably 20 different assays required just on the payload itself. Now you're surrounding this with at least 4 different lipids around it. Each would have their own purity, reactivity, compositional variability there. And of course, you're putting in this whole product inside a different buffers, stabilizers, etc. So now you have you probably have 20, 30 different components that you're looking at for any single product. Compare that to a small molecule where you have one often. So that's the first major problem.
The second major issue is how you handle the materials.
You've taken a nice soluble payload like mRNA in aqueous and water and surrounded it by insoluble lipids. So now you have to be careful how you handle it. Existing techniques that work well for mRNA don't work for an LNP because, again, you have basically an insoluble bunch of lipid drops.
And the third issue has to do with stability.
mRNA is inherently very unstable. The lipids we use some of them have different oxidation potentials. So you have to be very careful on the type of handling you do with it. You can't leave this on the bench for multiple days. You can't, even storage at 4 degrees is difficult with a lot of LNPs. So there's a limited time you have to analyze these materials. So that allows us to really have a handle from everything from the material chemistry, hardcore chemistry stuff, all the way up to the in vitro and vivo potency. And we've tried to fill every little dynamic in there that we can to meet our clients' need.
Robust analytics then aren't just academic, they really save time and help drug designers and drug makers actually move faster to the clinic.
Exactly. And Adam has a saying he likes to use, and that is that 2 weeks of analytics can save 2 months of development. We like the sound of that.
If you are having a problem, usually you see a problem as your final drug product not giving the potency or not giving the effect that you want. Analytics allows you to do that. You can say, hey, this material, this payload was degraded, so we need to prevent degradation next time. Or the final composition was not what we thought it was. We need to adjust it. There's a lot of different attributes we can measure that give us a very clear idea of what the cause of a loss of potency was.
All of this really shows why having strong analytics upfront is so critical.
Let's talk about multi-payload LNPs. There's a different challenge here. What is it?
The original LNPs, the first generation, had small oligos in them. Antisense oligos, siRNA, etc. Those had a million options for analysis. They were easy. There's HPLC, mass spec, all these things. Then we went to the mRNA vaccines, which had some techniques, fewer than 600 small oligos, but you could do things like size-based separations of the payloads. That generation we're starting to see, I'm not going to say come to an end, but certainly diverge into what's called the new modalities here, where we're seeing 2, 3, 4 multiple different modalities inside. And the problem there is that when you have the traditional ways of separating based on size, That doesn't work if you have compounds of the exact same size. And you see this a lot in the case of Cas9 applications. So the Cas9 systems, the CRISPR-based Cas9 systems, use a guide RNA to target the Cas9. But in a lot of cases, people are using two guides, one to cut upstream and downstream of where they're trying to manipulate. Typically, it's in the case where they're trying to do knock-in or knock-out experiments for gene editing. Of course, if you have two guides that are different sequences but exactly the same mass from a chromatographic perspective, you can't use size-based separations, and you need to have other tools to do this. And this is really where we started to find a need to support our clients to develop new tools that allowed them to provide the analytics to measure each of those payloads independently so that they could eventually go to qualification and go towards the clinic.
According to Adam, targeted LNPs are the next frontier.
99.9% of LNPs end up in the liver if you inject them intravenously. So everyone's focused on extrahepatic delivery. And there's different approaches people take. There's changing the ionized lipid compositions, and there's some encouraging data there. But there's also this idea that if you put a targeting moiety or targeting ligand on the surface of it, most commonly an antibody, it can pick out specific cell types, tissues, and focus it there. This is obviously ideal if you're doing, for example, we're seeing, especially in the last couple of years, a huge uptake in targeting. The problem, though, is now you have a complex LNP, and quite often you're putting a complex protein on the surface, and you're having to conjugate them chemically, and then analyze that whole thing together. So you have complexity plus complexity, which is great for me. I love that stuff, but it's not always the easiest.
And what's next?
We are starting to see a shift away from the early generation particle characterization techniques like dynamic light scattering. And we're seeing a lot of different technology. The one I'm most excited with is called AF4 or FFF mols, which allows us basically to do chromatographic separation of LNPs. That, I think, is going to be rather revolutionary in the industry, where we can understand the complexities across the different heterogeneity inside particles. So I think that there's a lot of interest coming there.
Wow, what a journey, Dodi, from Stephan's work at the Max Planck Institute on synthetic guide RNAs to Adam's cutting-edge analytics for lipid nanoparticles, making sure that we get these tools where they need to go safely and effectively in the human body so they can have the effect that we want them to have. Amazing. That's
right, and both of our guests are pushing the boundaries of gene editing, either in the lab or in the clinic. And that shows us how much precision, creativity, and teamwork it takes to make therapies a reality.
I guess now, as we think about the future of gene editing, as it unfolds from designing the guides to analyzing their delivery, it might be the right time for us to move to every day as a school day. What have you learned this week, Dodi?
I learned something. Actually, Connor, you've lived in the tropics, and I wonder, have you done...
Don't I miss it at the 99th right day of January?
Snow on the ground. Have you eaten insects? I have
eaten insects.
And what do you think of your dining experience with insects?
They taste like any crustacean. You get it right. Most things taste like chicken.
So... Or just spice. They just taste like spice.
Yeah. We shouldn't be icky about it. Most of the world eats insects. Those of us in the global north, in the US and in Europe, should stop being icky about insects. It's a great source of protein, so we should just get with the program.
And so the paper I read is really about digging into, what exactly are the advantages of eating insects, whether it's nutritional or for the climate. And these two scientists from the University of Montreal published their study in The Conversation, which is a great source for scientific studies. But it was republished for idiot minds like mine on phys.org. phys.org. And these two nutrition and food systems researchers looked at how emerging food sources, like edible insects, stack up nutritionally and environmentally. And the sum of the story is that, of course, meat production has this massive environmental footprint, taking up about 80% of agricultural land and generating about 12% of global greenhouse gas emissions. Are insects as efficient, nutritious, and sustainable as advertised? They also looked into, are insects the right new protein source to use fewer resources without compromising nutrition? And you can guess that the result is pretty nuanced. I mean, yes, insects are resource efficient, but the nutritional quality varies widely. So their overall recommendation is, be careful. They sound like a magic fix, but the reality is pretty nuanced. It depends on farming methods. It depends on the species of insect that you are dining on. But indeed, they are super resource efficient compared to traditional meat, which take up a lot of land, water, and feed. So I thought that was interesting to actually track back what's the real truth about eating bugs.
And what's so fun is it's going to so contribute to our lunchtime conversation in the canteen here at Cytiva.
Yes.
This week we had bioengineered synthetic meats, not on the menu, but certainly in conversation. So it's been a journey just of seeing the ick factor on people's faces as we talk about it.
Right, when you bring it up.
It's absolutely huge.
Yeah. What did you learn?
I learned that you just cannot trust a synthetic AI agent to generate a meaningfully accessible and believable Discovery Matters Every Day as a School Day segment if you don't train it properly.
This is Connor bringing the downside of AI. Usually you're so pro AI.
Yeah, it's just not happening here. I am 9 minutes into coaching the agent to find me something meaningful that I've learned today. We've now found ourselves buried in a story about living material engineered bacteria that do self-healing. I'm none the wiser, and I have significantly failed in my attempt to use AI to boost my knowledge gathering. So there, folks, is a warning for you all. Yes, AI can really help, but it only helps if you think very thoughtfully about what you're trying to get it to do for you. Sometimes you fail.
We celebrate that. That's okay to fail.
It is okay to fail, so I'm going to go back, retrain this agent to give me some better insight on a regular basis, and next time we come to Every Day as a School Day, I'm going to knock your socks off.
Our producer, mixer, and sound magician is Tom Henley. My name is Dodie Axelson.
And I am Connor McKechnie, recently humbled by AI. Make sure you rate us on Spotify or whichever platform you use. Thank you for listening to Discovery Matters, brought to you by the fun people at Cytiva.